Okay, so how does evolution actually build something new without totally breaking what already works? It's a great question. Well, we're about to uncover this secret recipe that nature has been using for, hundreds of millions of years. It's this amazing story of how life can be both a master at conserving what's important and a relentless innovator at the same time. And we're going to see it all through the history of just one single family of genes.
So, our story really kicks off with this genetic puzzle, a real head scratcher that had scientists stumped for a while. And it all starts when you take this same type of protein and compare it in two animals that, honestly, could not be more different. So, on one side, you've got this absolutely massive, complex protein in a fruit fly, the OVO protein. We're talking over 1,300 amino acids long. But then, on the other side, you have the OVOL1 protein from a mouse, and it's tiny. It's way simpler, less than a quarter of the size. So they're supposed to be in the same family, but they look just dramatically different. And that right there is the central mystery we're going to dig into. See, both of these proteins do a really similar job. They act like these key switches during development, turning other genes on and off to, build an animal. So the big question is, how can two related genes doing basically the same job end up creating proteins that look so incredibly different? All right, so like in any good detective story, the very first clue comes when you look a little closer at what they actually have in common. You've got to ignore all the differences for a second, cut through the noise, and find that one signal, that one piece that connects them across all that time. And what do you know? It turns out that buried deep inside both of these super different proteins is this identical little piece of machinery. It's called a zinc finger motif. The best way to think about it is like the protein's toolkit. It's like a set of tiny molecular hands that let it literally grab onto DNA and get its job done. And this right here, this is the proof. What you're looking at is the blueprint, the sequence pattern for those four zinc fingers. This is the engine of the protein. And this exact design is so essential that it's been kept almost perfectly. I mean, perfectly, from ancient sea anemones all the way to you and me. It's like the family secret passed down for hundreds of millions of years. Okay, so if the core engine is the same in both, then where in the world does all that extra size in the fruit fly protein come from? Well, this is where the story gets really wild. Let's get to our second and honestly more surprising clue. The answer is in everything that's outside of that core. Just take a look at these two graphs. The one for the mouse protein on the left is, mostly flat. That tells us it has a really stable, orderly structure. But now look at the fruit fly protein on the right. Whoa, it's this chaotic jumble of high peaks. Scientists have a great name for this. They call it intrinsically disordered. It's this beautiful chaos that evolution has just bolted on to that stable little engine. Now, it's really tempting to look at those messy, chaotic parts and just think, oh, that's junk DNA or something. But it's not at all. These parts are actually evolution's raw material for innovation. The Lego analogy is perfect here. It's like taking a standard, reliable Lego brick, that's our core, and then just snapping on all these new, custom, weird-shaped blocks to see what you can build. And the data right here, this proves it. This chart is measuring genetic diversity. So look at box A. That represents our stable, conserved core. See how narrow that range of diversity is? Not much changes there. Okay, now look over at box B. That's the chaotic part. The box is way taller, way wider. It shows so much more diversity. This is the smoking gun right here. These disordered regions are evolution's playground, where things are changing and innovating way faster than the stable core. So the really crucial point here is that this wasn't some one-time accident. As the researchers who figured this out said, this process happened over and over and over again in different animals. Evolution's strategy is simple. Keep the essential engine, those zinc fingers, and then in totally different animal groups, independently bolt on these fast-changing experimental parts. And that's how you get new functions. Okay, so now we have our two big clues, right? We have a conserved core, and we have these experimental add-ons. With these pieces, we can actually start to trace the family history of this gene. And not just for a few generations. We're talking across the vast expanse of deep time. The first thing we find when we look at this history is actually a disappearance, a mystery. If you look at this chart, you can see the OVOL1 gene and its neighbors all lined up nicely in mammals, amphibians, and fish. But just follow those lines over to the birds and reptiles, and you see a gap. The entire neighborhood of genes was just completely lost. Poof, just gone. So next up, researchers looked at OVOL2, which is another member of this gene family. And here, the story is totally reversed. This gene is there in most land animals, but as you can see, it completely vanished from the fish lineage. It's just another amazing example of how different branches on the tree of life are constantly pruning and customizing their own unique genetic toolkit. And here, we see a totally different strategy at play. In fish, the OVOL3 gene got duplicated. Think of it like a classic copy and paste error in a document, and then the original copy was lost, but the new one was kept. Now, this is a really powerful move in evolution because that new copy is now free to change and maybe take on a completely new job. Okay, so what does this all mean? We've looked at protein structure, we've looked at how fast different parts evolve, we've even seen genes get lost and duplicated. So why does the story of this one little gene family matter so much for understanding, well, all the diversity of life on Earth? Well, it shows us this really fundamental strategy, a playbook that's been playing out for over 700 million years. It all started with this simple core gene in some ancient animal. Then, around 400 million years ago, a bunch of major gene duplication events happened, and suddenly vertebrates had a bunch of copies to play with. And that brings us to today, where each group of animals has customized its own set, adding and subtracting these experimental parts to fit their needs. And honestly, this one single diagram just puts the entire puzzle together so beautifully. Look at the base of the tree. You've got a simple ancestor with just the core engine. But then, as you move up all the different branches, you can literally see nature independently bolting on new, highly variable pieces. Those are all the different colored boxes. The core stays the same, but the add-ons are totally unique to each lineage. So this is evolution's secret recipe for building all this amazing complexity. And it's so simple and elegant when you think about it. Step 1, preserve the functional core. Keep what works. Don't mess with the reliable engine. Step 2, innovate on the periphery. Attach these rapidly evolving, flexible add-ons to that core, creating new functions and generating all sorts of diversity without breaking the original machine. It's just such a powerful mechanism for generating the incredible diversity of life we see all around us. And it leaves us with one last, really fascinating thought. This process, it didn't just stop millions of years ago. It's happening right now in every single species on the planet, which leaves you to wonder, what new biological tools is evolution building as we speak by adding these novel extensions to ancient stable genes?